Let us now consider the fat-soluble vitamins. The four fat-soluble vitamins are designated using the letters A, D, E, and K. A, D, E, and K. Many of the functions of the fat-soluble vitamins involve processes that occur in cell membranes. The structures of the fat-soluble vitamins are more hydrocarbon-like, with fewer functional groups than the water-soluble vitamins. Their structures as a whole are non-polar, which enhances their solubility in cell membranes. Vitamin A, normal dietary intake provides a person with both preformed and precursor forms. Preformed vitamin A Preformed vitamin A forms are called retinoids. The retinoids include retinal, retinol, and retinoic acid. Foods derived from animals, including egg yolks and dairy products, provide compounds such as retinyl esters that are easily hydrolyzed to retinoids in the intestine. Foods derived from plants provide carotenoids, which serve as precursor forms of vitamin A. The major carotenoids with vitamin A activity is beta carotene, which can be cleaved to yield two molecules of vitamin A. Beta carotene is a yellow to red-orange pigment plentiful in carrots, squash, cantaloupe, apricots, and other yellow vegetables and fruits, as well as leafy green vegetables, where the yellow pigment is masked by green chlorophyll. Vitamin A has four major functions in the body: vision, regulating cell differentiation, maintenance of health of epithelial tissues, and reproduction and growth. Vitamin D. The two most important members of the vitamin D family of molecules are vitamin D3 or cholecalciferol and vitamin D2 or ergocalciferol. Vitamin D3 is produced in the skin of humans and animals by the action of sunlight or UV light on its precursor molecule, the cholesterol derivative 7-dehydrocholesterol, a normal metabolite of cholesterol found in the skin. Absorption of light energy induces breakage of the 9-10 carbon bond. A spontaneous isomerization, shifting of double bond, then occurs. So this is where you have the 9-10 carbon bond. So this is the shifting of the double bond. Only a few foods, including liver, fatty fish such as salmon, and egg yolks, are good natural sources of vitamin D. Such vitamin D is vitamin D3. Foods fortified with vitamin D include milk and margarine. The rest of the body's vitamin D supplies are made within the body, particularly the skin, with the help of sunlight. The principal function of vitamin D is to maintain normal blood levels of calcium ions and phosphate ion so that the bones can absorb these ions. Vitamin D stimulates absorption of these ions from the gastrointestinal tract and aids in their retention by the kidney. Vitamin D triggers the deposition of calcium salts into the organic matrix of bones by activating the biosynthesis of calcium binding proteins. Vitamin E. There are four forms of vitamin E: alpha, beta, delta, and gamma tocopherol. These forms differ from each other structurally according to which substituents are present at two positions on an aromatic ring. So referring to this position and this position. If you have in this position and this position both methyl groups, then you call it alpha tocopherol. If uh, in R prime you have methyl group and in R prime prime double prime you have a hydrogen, then that is the beta tocopherol. The tocopherol form with the greatest biochemical activity is alpha tocopherol. So this one. The vitamin E form in which methyl groups are present at both R prime and R prime prime or R double prime positions on the aromatic ring. Gamma tocopherol is the main form of vitamin E in vitamin E rich foods. Plant oils, margarine, salad dressing and shortening, green and leafy vegetables, and whole grain products are sources of vitamin E. The primary function of vitamin E in the body is as an antioxidant, a compound that protects other compounds from oxidation by being oxidized itself. Vitamin E is particularly important in preventing the oxidation of polyunsaturated fatty acids and membrane lipids. It also protects vitamin A from oxidation. Vitamin E's antioxidant action involves it giving up the hydrogen present on its hydroxyl group to oxygen containing free radicals. After vitamin E is spent as an antioxidant reaction with vitamin C, restores the hydrogen atom previously lost by vitamin E. A most important location in the human body where vitamin E exerts its antioxidant effect is the lungs, where exposure of cells to oxygen and air pollutants is greatest. Both red and white blood cells that are passed through the lungs, as well as the cells of the lung tissues itself, benefit from vitamin E's protective effects. Infants, particularly premature infants, do not have a lot of vitamin E, which is passed from the mother to the infant only in the last weeks of pregnancy. Often, premature infants require oxygen supplementation for the purpose of controlling respiratory disease. In such situation, vitamin E is administered to the infant along with oxygen to give antioxidant protection. Vitamin E also uh, it has been found to be involved in the conversion of arachidonic acid to prostaglandins. Vitamin K. Like the other fat-soluble vitamins, vitamin K has more than one form. Structurally, all forms have a methylated naphthoquinone structure to which a long side chain of carbon atoms is attached. This is the naphthoquinone. It is methylated. This is the methyl group. This is where the long carbon chain is attached. The various forms differ structurally in the length and degree of unsaturation of the side chain. Vitamin K1, also called phylloquinone, has a side chain that is predominantly saturated. Only one carbon-carbon double bond is present. It is a substance found in plants. Vitamin K2 has several forms called menaquinons, with the various forms differing in the length of the side chain. Menaquinone side chains have several carbon-to-carbon double bonds, in contrast to the one carbon-carbon double bond present in phylloquinone. Vitamin K2 is found in animals and humans and can be synthesized by bacteria, including those found in the human intestinal tract. Typically, about half of the human body's vitamin K is synthesized by intestinal bacteria, and half comes from the diet. Vitamin K is essential to the blood clotting process. More than a dozen different proteins and mineral calcium are involved in the formation of a blood clot. Vitamin K is essential for the formation of prothrombin and at least five other proteins involved in the regulation of blood clotting. Vitamin K is sometimes given to pre-surgical patients to ensure adequate prothrombin levels and to prevent hemorrhaging.